welcome you in my video. So for today's video, we'll be discussing about Marcello Malpighi's history, his inventions, and his discovery. Marcello Malpighi was born in March 10, 1628 and died on November 30, 1694. He was an Italian biologist and physician who is referred to as the founder of microscopical anatomy, histology, and father of physiology and embryology. Malpighi made a significant contributions to the field of anatomy and histology. Here are a few of his noteworthy inventions. First, microscope, then Malpighian tubules, next capillary network, then Malpighian corpuscles. So let's discuss two of his inventions. First, let's discuss the compound microscope. A compound microscope is a type of microscope that uses multiple lenses to magnify the image of a specimen. It consists of several key components that work together to produce a magnified image. Here are the parts of microscope. First is the eyepiece. The eyepiece is also known as the ocular lens. It is the lens through which the observer looks into the microscope. It typically provides a magnification of 10 times or 15 times, allowing the observer to view the enlarged image. Second, objective lenses. The objective lenses are located on the rotating nosepiece just below the eyepiece. These lenses provide varying levels of magnification, typically ranging from 4 times to 100 times or higher. The microscope may have multiple objective lenses, which with a different level of magnification. Third is the stage. The stage is a flat platform where the specimen is placed for observation. It usually includes clip or other mechanisms to hold the specimens in place. The stage can be moved vertically and horizontally, which using knobs to position the specimens under the objective lens. Next is light source. Most compound microscopes have an integrated light source located beneath the stage. This light source, often a LED or a halogen bulb, illuminates the specimens, allowing it to view more clearly. The light can be adjusted in intensity using a control knob. And last is the condenser. The condenser is a lens located beneath the stage that helps concentrate the light into the specimen. It plays a crucial role in enhancing the contrast and clarity of the image. So, when using a compound microscope, the observer positions the specimen on the stage and select desired objective lens. The coarse and fine focus knobs are then used to bring the specimens into the focus by adjusting the distance between the objective lens and the specimen. The observer looks through the eyepiece to view the magnified image. As light passes through the specimen, it interacts with the various structures and components, causing them to scatter or absorb the light differently. The variation in light is then captured by the objective lens, which magnifies and projects the image onto the eyepiece. The eyepiece further magnifies the image, allowing the observer to see fine details that are not visible to the naked eye. By combining the magnification of the objective lens and eyepiece, compound microscopes can achieve high levels of magnification, enabling detailed examinations for microscopic structures. Now let us proceed to his discovery. The capillary network, also known as capillary bed. It is a network of tiny blood vessels that connects arteries and veins. It is an essential part of the circulatory system, responsible for the exchange of nutrients, gases, and waste materials between the blood and the surrounding tissues. Capillaries are the smallest and the thinnest blood vessels in the body, with a diameter ranging from 5 to 10 micrometers. They consist of a single layer of endothelial cells, which are cells that line in the inner surface of blood vessels. Capillaries are so small that blood vessels, such as red blood cells or RBC, have to pass through them in single file. The capillary network is highly extensive, with billions of capillaries distributed through the body. This extensive network ensures that every cell in the body is in closest proximity to a capillary, allowing for efficient exchange of substances. The main functions of capillary network is to facilitate the exchange of substances between the blood and the surrounding tissues. Oxygen and nutrients, such as glucose and amino acids, are delivered to the tissues by the blood. These substances diffuses across the thin walls of the capillaries and enter the interstitial fluid, which surrounds the cell. At the same time, waste products such as carbon dioxide and metabolic byproducts diffuse out of the cells into the interstitial fluid. These waste products are then carried away by the blood through the capillaries and transported to organs responsible for their elimination, such as the lungs or kidneys. The exchange of substances across the capillary walls occurs through several mechanisms. This includes diffusion, which is the movement of substance from an area of higher concentration to an area of lower concentration, and facilitated diffusion, which involves the use of carrier proteins to transport specific substances across the capillary walls. In addition to nutrient and waste exchange, the capillary network also plays the role in regulating blood flow and blood pressure. The diameter of the capillaries can be influenced by various factors, such as local metabolic activity and release of certain chemicals. This allows for the redistribution of blood flow to areas of higher need. Overall, the capillary network is a crucial component of the circulatory system, enabling the exchange of substances between the blood and the tissues and ensuring the proper functioning of the cells throughout the body. So, these are the history, the discoveries, and the inventions of Marcello Malpighi. But I only included one discovery and one invention. The microscope for the invention and the discovery is the capillary network. So I hope you enjoyed the video and you've learned a lot. Thank you for watching.